everyone, and welcome to today's webinar titled Innovative Tools for Efficient CRISPR Cas Genome Engineering. This webinar is part of the 5th annual CRISPR virtual event, and I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by IDT. For more information about IDT, go to idtdna.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the left of your screen. Now, you can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. Now, if you have any trouble seeing or hearing this presentation, just click on that help desk button located at the top of your screen with the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract tab from the menu on the left and use the screen and follow the process to obtain those credits. I now present today's speaker, Dr. Gavin Kurgan, a Bioinformatics Application Manager at Integrated DNA Technologies. For a complete biography of our speakers, please visit the Presenter tab from the menu on the left of your screen. Dr. Kurgan, welcome. You may now begin your presentation.
Thank you for the introduction. Just want to start off and say thank you to Labroots for putting on the 5th annual CRISPR 2022 event. I'm really excited to speak with everyone today and share some of the work that we've been doing at IDT. So briefly, my name is Gavin Kurgan. I'm a Bioinformatics Applications Manager here at IDT. And today I kind of put together a pretty broad topic CRISPR talk to discuss a lot of the innovative work we've been doing at IDT and how that impacts a number of different CRISPR-Cas workflows. So as a quick review, the main enzymes in the genome editing toolkit that we provide and that are often used to edit mammalian cells are usually Cas9 and Cas12a. These two enzymes have distinct mechanisms, structural requirements, and different levels of specificity. But using either enzyme, a double-strand break is generated at a targeted location in the genome, which the cell now needs to repair. This is done using the cell's endogenous DNA repair pathways. And the specific preferential outcomes here can really vary from cell type to cell type. But generally speaking, there's two main mechanisms that are leveraged by researchers to generate mutations in the resulting cell population. When no donor DNA template is provided, the cell predominantly undergoes non-homologous end joining, which can generate variable insertions, deletions, and substitutions adjacent and overlapping with the site of the double-strand break. Alternatively, if a DNA donor template is provided with homology arms targeting the regions adjacent to the double-strand break, shown here in blue, specific mutations can be incorporated into the resulting population of cells using the homology-directed repair pathway. However, a key finding of CRISPR genome editing, like many other oligotherapeutics, is that the targeting of these enzymes is imperfect and may induce off-target unintended double-strand breaks at similar genomic loci. Here you can see the output of an empirical off-target nomination tool, GuideSeq, for a guide targeting the EMX1 gene. The number of reads of this workflow generally correspond to the prevalence of these off-target editing events. And here you can see that the on-target site with the most reads at the top and some of the identified off-target loci and the respective mismatches to the targeting sequence shown below. This is just one method of many for nominating off-target sites, but the key takeaway here is that off-target double-strand breaks occur, and identifying and quantifying their impact is important. And so all of this is pretty much a primer. Like I said, this is going to be a pretty broad presentation and kind of topic, but my goal here is to try and help connect all these different pieces of the genome editing toolkit and how those tools are currently being innovated on here at IDT. Here you can see a broad range of our product portfolio, starting with design tools to help create oligos and perform and interrogate genome editing, the reagents and proteins that need to be coupled with these oligos to make them functional, the broad swath of different enhancers and oligotypes for enhancing targeted DNA repair. And then finally, the tools provided to actually detect genome editing experiments. And we'll walk through each of these throughout the presentation. So first, we'll cover considerations in design of both guides and donor oligos for HDR and the IDT tools that exist to help perform these tasks. So guide RNA design is one of the most important first elements of experimental design. This requires you to ask what region of the genome you are targeting, what kind of PAM or enzyme requirements you have, if you're going to be leveraging highly specialized guide RNAs for introducing edits, and what kind of metrics should be optimized in guide selection. From here, people can leverage existing IDT tools to design guides for both standard and custom applications, design large arrayed functional targeted libraries, or design guides with the intention of introducing an edit through the repair pathways like homology-directed repair. First, we'll actually just discuss the basics of how tools through the IDT portal can actually be used to perform both standard and custom guide RNA design and synthesis, though. For Cas9 guide RNAs with an NGG PAM, we have a full guide RNA design tool that supports most popular genomes and generates both on and off-target scores to help prioritize which guides are best for a desired region. It can then be relatively easily funneled into a lot of our standard, two-part or sgRNA products and can be ordered in bulk. However, many people may use custom nucleases or guide RNAs that are highly specialized, like PEG RNAs for prime editing. For these applications, we have a custom guide RNA ordering tool that can accept lengths from 30 to 150 nucleotides, and many of our standard modification patterns, all in a single submission or in bulk formats. This includes, basically designing for guides like Cas12a, Cas13, like I said, PEG RNAs, and more. All you really need to do is have means to design the sequence, which can be done through several specialized open source tools. And it should be noted, we can synthesize guide RNAs longer than our standard 150 nucleotides. However, these are evaluated kind of on a custom ordering process to make sure that the synthesized product can actually be held to IDT quality standards. When someone wants to generate a large functional screen of the genome, though, this doesn't often fit easily into the standard ordering processes, especially because it requires special design considerations and ordering mechanisms. For that, we have a special service for accepting guide RNA library designs. Currently, we support design of synthetic arrayed CRISPR libraries. For those new to the subject, these are typically introduced into experimental paradigms where the goal is to screen a large number of genes for phenotypic outcomes in high throughput. For example, you may have a plate of cells stably expressing Cas9 or that are prepared to be co-transfected with an RNP, along with this arrayed guide RNA library you're creating. These libraries are designed to typically have multiple guides per transfection that are all targeting the same gene in one well. From there, there is a high level of functional knockout typically observed, which makes this plate of edited cells ideal for screening to identify gene candidates of interest, which I'm indicating is positive hits here with yellow stars. One thing that we have worked a lot on recently is optimizing how these are done so that we can provide an optimized design and synthesis service for performing guide RNA library screens. For starters, we looked into just how many guide RNAs are actually needed in a well to get really robust editing across 96 different genomic targets using our guides and design algorithm. This led to our recommendation to try and design 3 guide RNAs per gene target for robust editing, but also enabling 2, which still allows very high levels of editing, as seen on the left. Then we perform designs for this type of experiment. To do this, we created an automated tool to look up exon usage patterns in annotated and canonical transcripts and look to target exons which will disrupt the most common transcripts as early as possible in the shared targetable exons. From here, we look to place highly functional guides within a region interrogatable by amplicon sequencing to enable downstream genotyping with Illumina sequencing. And all of this can kind of be seen in that graphic in the middle. We've included a number of other optimizations as well, where our design guides are actually selected to attempt to optimize frameshift frequency by targeting certain genomic context and sequence repair features that we're aware of. Here you can see that on the right, where the currently utilized strategy, highlighted in green on the left, outperforms designs where only on-off target information is used, which is highlighted in orange, to prioritize guide RNA selection, as is done in many other algorithms. However, our designs are handled on a custom process and can accommodate customer requested changes as required and can be reached using the link at the bottom of the slide. Lastly, when guides are being designed to ultimately introduce targeted changes through homology-directed repair, this requires specific optimizations that we recommend go through our HDR Design Tool. Some of the reasons why it is so important to consider guide RNA design simultaneously with HDR donor design can be seen with these two graphics. For one, we know that HDR efficacy is greatly affected by the relative location of the guide RNA cut site to the intended introduced changes. Here, as you can see, HDR indicated on the left graph's left axis, and the distance to the cut site to an introduced ECO-R1 insertion via a donor on the x-axis. As distance from the cut site increases, HDR efficacy drops, especially without the introduction of what we call track mutations, which are non-coding mutations that are introduced along the way to an intended change to attempt to favor complete introduction of donor mutations instead of partial introduction. Secondly, we can see on the right, when we consider targeted mutations that are not near a cut site, the strand used to introduce donor-guided mutations is particularly important, with non-target strand donors being more effective introducing mutations 5' of the Cas9 cut site, whereas target strand donors are more effective in introducing mutations 3' of the cut site. As distance increase, if we introduce these track mutations that I mentioned earlier on the correct strand, high levels of HDR can still be maintained, as indicated by the track annotation on the x-axis on the graph on the right, with the y-axis indicating the perfect HDR frequency relative to an insertion right at the cut site. Following this work, we realized it would be good to have a design tool that considered guide information in the context of what donors could introduce a desired mutation. This led to the creation of our Alt-R HDR Design Tool. The Alt-R HDR Design Tool is an interactive web-based tool reachable at the link at the bottom right that makes it a lot easier to consider the need to co-optimize HDR donors along with their respective guides and order both. It allows you to come with a number of different inputs types to help specify the mutation you're interested in that you would like to create and adjust experimental parameters like homology, arm size, the use of silent mutations, and more. Many of these parameters come with intelligently defined defaults as seen in one of our recent publications. But you can always come with your preferred guide RNA or search for the best guide RNA given your mutation of interest. I think it's always pretty hard to explain interactive tools and slideshows, but I would just highly recommend checking it out for your next experiment. It also allows you to preview your translation before and after the theoretical edit to make sure you're really making the functional change that you hope to. Next, I'll switch to discussing the different enzymes and formulations you can choose in performing CRISPR Cas genome engineering. IDT offers several different enzymes for genome engineering with quite a few IDT exclusive mutants, which are highlighted here in orange. We offer both Cas9 and Cas12a enzymes, which allow for editing based on the presence of NGG or TTTB PAM sequences near your desired editing location. For Cas9 nucleases, we offer both wild-type and hi-fi Cas9 that was developed using bacterial mutagenesis. The original AS and LB enzymes for Cas12a also have issues with low on-target editing, so we engineered higher activities of both of these different enzymes. Finally, we have fluorescently labeled Cas9 options like Cas9 GFP and RFP that allow for visualization of delivery, although I won't talk in great detail about those today. Wild-type enzymes can have significant off-target editing depending on what guide and delivery method is used. Here we can see just how important quantifying off-target editing effects is for an AAVS1 targeting guide and 28 nominated off-target loci that we evaluated in-house under variable experimental conditions. Here you can see the percent editing quantified by NGS on the y-axis, with each on or off target on the x-axis. Using a cell line with continuous expression of wild-type Cas9 complex, we observe a large amount of editing at the on-target and also highly prevalent editing at these 28 off-target loci. By simply switching to ribonucleoprotein nucleoffection, we can see that editing at these off-target loci is greatly decreased. This really emphasizes the importance of optimizing the delivery mechanism of your CRISPR-Cas complex for highly specific editing. Furthermore, by switching to a highly specific CRISPR-Cas enzyme variant, like our Alt-R hi-fi Cas9, these off-target effects can be further reduced while maintaining efficient editing at the on-target site. We recently published results on this Cas9 mutant in 2018 with the publication title listed below. This engineered variant was recently benchmarked independently in the creation of the 2020 TTISS method for profiling Cas9 endonucleases. Here, relative activity of different nucleases was tested with respect to activity as measured on the y-axis and specificity as measured on the x-axis, relative to wild-type Cas9. It can be seen here that our hi-fi Cas9 is one of the most specific engineered Cas9 variants tested that manages to still maintain comparable activity to wild-type Cas9, which is seen in the top left corner. A popular alternative to the Cas9 system is Cas12a, otherwise known as CPF1. This system uses a short single guide RNA and is natively highly specific. In contrast to Cas9, which recognizes GC-rich NGG PAMs, the Cas12a protein recognizes TTTB PAMs. It also is a smaller protein in size as compared to Cas9. As mentioned earlier, wild-type AsCas12a is not as efficient at editing the genome of human cells as other enzymes like SP-Cas9. So we undertook an effort to perform scanning site saturation mutagenesis, coupled to deep sequencing, to identify variants that increase genome editing activity. Here on the left, you can see the result of this screening from around 300 nucleotides to 1,300 nucleotides in the AsCas12a sequence before and after selection of beneficial variants. The peaks here indicate enrichment of certain mutations. This allowed us to identify the M537R and F870L mutations that make up our vastly improved AsCas12a Ultra enzyme. The graphic in the middle shows how the Ultra mutant greatly improves editing, as measured on the y-axis, at 96 different sites in HEC293 cells as compared to the wild-type AsCas12a nuclease. Additionally, performance of this variant has comparable editing efficacy as compared to SP-Cas9. Recent work that we collaborated with Editas Medicine on used this engineered nuclease in the editing of T cells and HSPCs, and saw that the benefits observed in HEC293 cells are transferable to more relevant cell types for genomic medicine workflows. While both hi-fi Cas9 and AsCas12a Ultra are excellent genome editing tools, other applications demand additional tools. Specifically in the field of agriculture, most editing is done in plants that grow and reproduce at lower temperatures. LB-Cas12a is an ortholog of AsCas12a that is known to function better in plants. However, similar to wild-type AsCas12a, it also suffers from poor on-target potency, and many of the mutations published for AsCas12a, including our own, are not directly transferable to LB-Cas12a. To this end, we undertook another effort to perform scanning site saturation mutagenesis of all positions in the LB-Cas12a protein. The best mutants from this library were stacked and tested using 4 different guides in human cell lines, initially as single mutation variants. From here, we found 16 mutants that were positive hits for increased activity and proceeded to combine the best mutations as double, triple, and quadruple mutants. The relative activity of these mutants compared to the wild-type LB-Cas12a are shown here on the y-axis, and the most universally improved stacked mutant was called LB-Cas12a Ultra, and is shown in this graphic with the red asterisk in the triple mutant category. Similar to our previous effort with the AsCas12a protein, the LB-Cas12a Ultra protein, intended for use in agricultural editing, demonstrates improved editing efficiencies relative to the wild-type protein. We demonstrated that here with collaborators from the University of Maryland with 6 TTTB PAM targets in plant protoplasts at low temperatures of both 25 degrees Celsius and 32 degrees Celsius. Here, the LB-Cas12a Ultra protein shows improved efficiency at all conditions compared to the wild-type protein. Now that we've talked about the guide RNAs and proteins used to edit the genome, we can discuss some of the different reagents and oligos used to drive editing towards intended genomic changes. As mentioned prior, after a double-strand break, the cell needs to repair DNA. Ideally, for intended genomic changes, we want the cell to utilize the homology-directed repair pathway to repair the break using an exogenously provided donor DNA. However, a number of different factors affect the efficacy of repair through HDR, including how the donor oligo is designed, the format in which it is provided, and even the cell cycle and transcriptional profile of the edited cells. Here we'll discuss the different options and optimizations that can be leveraged for this step. After Cas9 or your CRISPR enzyme of choice generates a double-strand break, ideally your donor will result in a nice clean repair through HDR. When using a single-strand DNA template, as shown here, this is the primary outcome, shown here on the left. However, there are additional misincorporation outcomes through NHEJ-based mechanisms that we'll address more in just a moment. When it comes to generating short insertions or SNPs, IDT offers our Alt-R HDR donor oligo product. These are modified oligos that are manufactured using our high-quality ultramer chemistry. They're low-cost options and are great, honestly, generally for generating these types of smaller edits. When it comes to large insertions, we have historically offered a long single-strand DNA template called megamers. However, these long single-strand DNAs can be challenging to make, which leads to higher costs, slower turnaround time, and greater limitations on the types of sequence complexities that can be accepted. To address some of these limitations for our customers, we began to consider options for double-strand DNA templates. Unmodified linear double-strand DNA, such as G-Block's hi-fi gene fragments, work well for HDR, but do have one major drawback. The blunt ends of the donor can also be recognized by the NHEJ repair machinery, leading to the possibility of homology-independent or blunt integration of the donor, shown here on the right side of the graphic. At the on-target double-strand break, this leads to the duplication of one or more homology arms. For long templates, this is generally a low-frequency event that can be screened out. However, this can also occur at endogenous double-strand breaks or at Cas9 off-target double-strand break sites. Unless you are looking for them, they could have unwanted effects downstream. So we asked the question, can modifications to the ends of the double-strand DNA mitigate the risk of this unwanted event? So we conducted an experiment with short double-strand DNAs with variable oligo modifications. Once we had identified top modifications from an initial screen, we then set out to further confirm them in additional cell lines and at additional sites. You can see the results here of testing variable modifications in 3 different cell lines at 5 different genomic loci. The y-axis here shows the average fold change of HDR, blunt integration, and the HDR blunt ratio compared to an unmodified double-strand DNA template for each modification on the x-axis. From this, we found that our top modification pattern, labeled as the Alt-R mod here, improved the ratio of HDR to blunt insertions at the on-target site 4-fold. This product is now available as our Alt-R HDR donor block product, which can go up to 3 kb. It offers faster, cheaper, and more flexible option to our long single-strand DNA product. Additionally, the modifications we've incorporated promote HDR while reducing unwanted blunt integration, shown here with the arrows. However, sometimes it is still desirable to further drive outcomes to HDR with additional approaches. An additive approach to promoting HDR is the use of either small molecules or peptides to modulate the transcriptional and functional activity of DNA repair pathways. This can dramatically improve HDR rates and additionally drive down unwanted repair outcomes. Our currently available product is the Alt-R HDR Enhancer V2, but I'll also give a sneak peek of one of our upcoming peptide products for improving HDR in this section. To look for small molecules that improve homology-directed repair outcomes, we performed a screen of a number of different small molecules, including many of those that had been previously published. The y-axis in this graphic indicates the percent perfect HDR, while the x-axis is indicating all the different small molecule treatments tested. This led to the initial identification of our HDR Enhancer V1, which is shown here in light green. Since then, though, we've continued screening and identified more compounds, and which led to the identification of our HDR Enhancer V2, shown here in dark green, which provides significant improvement for HDR rates even above the V1 enhancer. In addition to the best improvement in HDR efficiency, the V2 enhancer is also delivered at a lower concentration, which allows for better cell viability as well. Once we identified our oligo modifications and ideal small molecules for HDR, we then went to look at how it behaved with more difficult large knock-ins relative to alternative templates. Here in this graphic, we're comparing different repair templates when generating GFP tags at 3 different genomic loci. Without any additional treatment, we can see here on the y-axis that long single-strand DNA gives around 5% HDR as a baseline. Unmodified double-strand DNA gets up to about 9%. And Alt-R modified double-strand DNA boosts up to 15% HDR. We then use long-read sequencing to dig further into the editing and can see that the long single-strand templates actually result in a lot of partial integrations that are not observed as frequently with double-strand DNA templates, shown there on the right with the IGV plots. We then performed the same experiment in the presence of HDR Enhancer V2, and we're pleased to see that the treatment was very beneficial for the long double-strand DNA templates as well. V2 Enhancer brings single-strand DNA donors up to about 10 to 20% versus up to 50 to 70% with the Alt-R HDR donor blocks. As I said earlier, we also have a peptide-based HDR enhancer coming soon. There are a number of different ways to try and target DNA repair, but one is to potentially enable end resection to occur more freely. It's been published that the introduction of I53 improves HDR putatively by sequestering 53BP1, which is an inhibitor of end resection. We tested I53 ourselves, and while it worked when delivered as plasmid, the results were relatively inconsistent at different genomic loci when delivered as protein alongside Cas9 RNP. You can see those results here for one site where increasing concentrations of I53 protein are needed to see any sort of significant effect for HDR, represented here on the y-axis. Thus, we set out to engineer a ubiquitin variant with higher affinity for 53BP1 that would work better when delivered as a protein. From another site saturation mutagenesis effort, we identified a number of single amino acid changes that improved the affinity of I53 for 53BP1, shown here on the right as a fold increase in affinity versus the wild-type protein. A number of different mutants were generated, but two mutants with stacked mutations, named UBVA and UBVB, have vastly improved affinity. When testing these mutants as functional proteins, the improvement to HDR is increased above what we originally showed with wild-type I53 in red, but also was efficacious at significantly decreased concentrations of the actual peptide itself. We then wanted to test if our ubiquitin variants could be used as part of a cocktail with other HDR enhancers, like our E2 enhancer. Shown here is data looking at HDR rates using UBVA, Alt-R HDR Enhancer V2, or both in combination with different types of donors, including short single-strand DNA donors, AAV, and long double-strand DNA donors. We find that while the benefit of UBVA compared to V2 depends on donor type, we see an additional boost in HDR when they are used together. With the lack of observed toxicity, promotion of HDR by facilitating end resection, and compatibility with other HDR enhancers, we think that the ubiquitin variants targeting P53BP1 will be a valuable tool to facilitate HDR. Lastly, once transfection has been performed and cells edited, there is a need to quantify what the resulting effects were. We've shown a lot of data that has relied on that, but in this section, we'll talk a little bit more about how the tools we've created enable non-bioinformaticians to analyze NGS data for these purposes. When we talk about selecting optimal guide RNAs and quantifying their effects, this usually begins by selecting the best guides based on in silico predictions of on and off target editing efficacies, and verifying the guide's performance using a quick on-target editing activity assay. This is then used to select the most efficient guides, which then can have off-target effects nominated using an empirical method, like guide-seq or change-seq. From here, editing can be quantified at on and off-target sites simultaneously using a multiplexed PCR, which is possible using the rhAmpSeq system that I'll discuss shortly. However, when we talk about quantifying editing at on and off-target loci, there are several methods that currently exist, with variable levels of features as depicted in this graphic. One fast and cheap method is to perform a heteroduplex assay, such as using a mismatch endonuclease, such as the T71 or Surveyor systems. This method is fast and can be performed at high throughput, but is unreliable when it comes to quantification and provides no information on the allelic distribution of the edited cells. Here we can see the results from previously published work where a known concentration of edited DNA was used to evaluate the accuracy of these methods. We can see here that T71 and Surveyor systems quantification of editing efficiency is very approximate and only achieves a rough quantification of this target when 30 to 50% of the DNA is edited. Additionally, T71 and Surveyor systems can only quantify editing at one locus at a time. However, this is also highly dependent on the allelic diversity of the population of edited cells, representing yet another caveat that can lead to inaccurate quantification using this method. Sanger traces can also be used to approximate editing efficiency and the allelic distribution at a site using tools such as TIDE or ICE. Here we can see a graphic we adapted from Shapiro et al. That quantified the difference in editing efficiency using NGS represented on the x-axis or Sanger methods on the y-axis for a number of sites. Although we can see here that Sanger deconvolution methods generally correlate well with NGS results, they consistently underrepresent the editing efficiency of a population of cells. Additionally, the resolution on allelic diversity has previously been reported to be low for Sanger assays, and we can see that as the editing efficiency drops below 10% indels, these assays become erratic and unreliable. This is why we recommend that next-generation sequencing be used to quantify the effects of on- and off-target editing from CRISPR. Here we can see data derived from our recent publication where we evaluated the concentration of a titrated double-strand DNA mixture with a known concentration of indels in comparison to what is quantified using NGS. The values here correlate well with expectation down to 0.1% of observed indels, which is around the level of sequencing noise for this sample. NGS has high resolution of allelic variant distribution in the population and can be used to simultaneously quantify on and off target editing effects using the multiplex PCR action, like I said earlier. One caveat here, however, is that NGS analysis is really dependent on bioinformatics software, which requires accuracy and a lot of testing, really. Additionally, sometimes even just having access to bioinformatics expertise is a significant barrier. And this is a major issue that I'll show you how we solve with the rhAmpSeq CRISPR Analysis System. So the rhAmpSeq CRISPR Analysis System is a new end-to-end workflow we've developed to help researchers quantify the effects of on and off-target editing using NGS. This includes developing Rh primers for multiplex PCR using our rhAmpSeq Design Tool, preparing the amplicon libraries using the corresponding master mix, and analyzing the sequencing data produced from an Illumina MiSeq in an easy-to-use web platform. You can find more information on any of these compounds at the link below. Components, I should say. However, this workflow allows researchers to simply come with a list of off-target locations and convert this into high-quality NGS results without bioinformatics expertise. The rhAmpSeq CRISPR master mix comes with credits to do NGS analysis after sequencing. And the software for doing this analysis is specifically designed to be user-friendly and not rely on local computational resources. For the sake of time, though, I'm just going to focus here on the capabilities of the NGS analysis software that we call the rhAmpSeq CRISPR Analysis Tool instead of the whole portfolio. So you may be asking, why did we create our own CRISPR NGS analysis software and don't other open source softwares already exist for this? And the short answer here is yes, but based on our own findings and surveyed feedback, it doesn't really satisfy all end user needs. These tools currently consist of software like Crispresso, Crispresso2, Amplicon, but we found a number of customers struggling with these tools. The tools can either be difficult to use for those without bioinformatics expertise, or the accuracy of the actual software has not been fully characterized and validated. And a lot of these interfaces that exist for all of these softwares don't really support high throughput functionality and high throughput workflows. For this, we created our own tool by performing several Cas9 and Cas12a editing experiments and characterizing the resulting indel profiles of these experiments. We use this to create a biologically informed, thoroughly verified software package for CRISPR genome editing with an easy-to-use web interface. Here you can see how our tool in green outperforms other tools in annotating editing efficiency in an internally created in silico benchmarking dataset with a known amount of indels represented as a black line in the graphic. From a high level, the analysis workflow is as follows. You first upload files using either a drag and drop interface or by picking a folder on your hardware that can be used to stream the files into the interface. Next, you select your uploaded samples and provide guide and amplicon information to start an analysis run. Lastly, you can interpret the results of individual or aggregated experiments directly in the interface to quantify important metrics like editing, HDR repair frequency, frame shift frequency, the population of alleles, and more. Finally, we wanted to see how sensitive the rhAmpSeq CRISPR analysis system may be for quantifying low levels of off-target editing. We thus created a truth set with variable levels of indels spiked into 20 unedited genomic loci with background indel frequencies in our expected range between 0.0 to 0.4% indels. This enabled us to create a set of paired treatment and control sets that could be categorized as true positives when a control sample was compared to a treated sample, or a true negative when a control sample was compared to another control. To provide a binary call if a sample was edited or not, we performed a Fisher's exact test on the paired samples, comparing the read count of wild type and indel-containing reads for the assigned treatment versus control. This allowed us to generate a p-value, which we then used as a threshold to qualify anything with less than 0.05 as a truly edited site. Here you can see with about 47 to 56 pairs per indel dilution level, we are able to correctly characterize 95% of true positives down to 0.5% indels in this data set, which while correctly characterizing true negatives, approximately 95% of the time. A key requirement of this was performed with at least, that this was performed with at least 5,000 reads per target, paired treatment controls, and background indel levels less than 0.4%. And that really concludes a lot of the newest innovations and tools from IDT that I wanted to share with everyone here. Lastly, if you want more general information about rhAmpSeq and its applications, please feel free to follow this link. And with that, I'd just like to say thanks for tuning into this webinar, and I'll take any questions the audience may have.
I want to thank you, Dr. Kurgan, for your time today and for your important research. And before we go, I want to thank our audience for joining us as well today. I'd also like to thank our sponsor, IDT, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until November 2nd, 2024. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.